Let me introduce our next speaker, who bookends Tamara's story. You heard from Dr. Schoenberger how the neoantigen platform was developed, and now you'll hear from Dr. Miller about its implementation and execution. Dr. Aaron Miller was a fellow here in hematology oncology. He received his PhD in immunology at Northwestern and his MD also at Northwestern. Okay, thanks, Aaron. We were lucky enough to recruit him here for fellowship, and even luckier to recruit him here as a junior faculty member. He has been working with Dr. Schoenberger at La Jolla Institute in his laboratory, and has really been the driver of making the vaccine a reality. The second smartest person that I know. So without any further delay, Aaron, come to the podium. Thanks.
Thank you. Can you guys hear me? Can you hear me now? All right, bueno. UCSD is like the mafia. Once you're in, they never let you leave, right? So I'm happy to be here, and that was really powerful to hear Tammy talking about her experience with this. I'm honored to have been part of her treatment team and really taking this preclinical science that's been developed through this joint effort between Moores Cancer Center and the La Jolla Institute and just translating it, making it a reality. You know, this was conceptualized 5 years ago, and where we've come from then, early in my career, I think it's just an incredible thing to be part of, and we're going to carry on with this. So thank you, Ida, for the opportunity to speak today. This is a wonderful forum. It does take about a year to get this together, so it's wonderful to see this great of a turnout and to facilitate conversation. So a big motivation for my decision, my intent to pursue a career as a physician scientist, and specifically in the field of immuno-oncology, is how frustrating it is to see a survival curve like this. So earlier this morning, I was down in clinic. You know, for those of us that are both clinicians and scientists, we're presented with patients with cancer, devastating diagnosis, stage 4 disease, where at the point of diagnosis, 100% of patients are alive. They come to see you. They're scared. They want to know what their options are. But invariably, for patients with metastatic disease, at some point they would succumb to the disease. And we're trained, and we have a lot of research and development, be trying to push out this curve to improve progression-free survival, to improve overall survival. And it can work. The emphasis being quality of life or living with cancer, knowing that the treatments can be incredibly difficult to tolerate. Now, within the last decade, really, a reason that I'm in this field, and I think a lot of people in this room, is that immunotherapies have really shown that they can change that script. Increasingly, they're a new cornerstone of how we treat and how we approach cancer. Original studies with James Allison for metastatic melanoma, where life expectancy was about 8.5 months. Drugs like ipilimumab, you're seeing complete responses. And some of those patients are 10, 15 years out with no evidence of disease. That is a real testament, an aha moment saying this biology is real, despite our best efforts as scientists and clinicians to mess it up or not understand why this is occurring. It is a testament that it can work. And part of our goal is to take these tails, these survival curves that are now manifesting themselves, and recognize that there is really room for improvement to understand this better through basic science, to translate it, and to really improve patients' quality of life with ideally less toxic therapy. So I think this audience is very well versed that there are general approaches to immunotherapy. We have immune checkpoint blockades, we have TIL therapy, CAR T, TCR-T for engineered T cells, and NK therapies, all of which are being investigated, and all of which show promise in this field. What Steve talked to you about earlier is our efforts of neoantigen discovery. So neoantigens are, we think, very important. It's just really difficult to find the right ones, and to find a multitude of them. So through this joint effort between La Jolla Institute and Moores Cancer Center, this HLA-agnostic approach of functionally testing, don't guess but test, allows us to not predict what we think are relevant neoantigens, but actually to validate them. And not just to validate them, to have a multiplicity of them, more shots on goal, more targets against the patient's cancer direct a personalized cancer vaccine. A lot of it we learned from the fuzzy test tube with mice. Part of the latest ARM that Steve had alluded to with poly-IC, more doses, the combination with Keytruda, that research at the bench has literally been translated into amendments to this trial to make it better. So let's talk about what we're doing for the personalized cancer vaccine trial. So this is a phase 1b safety and feasibility study of these personalized immunotherapies, vaccines. So this was the first iteration of the trial, where you'd have a patient that comes into the cancer center, and like most trials, we're doing our screening for eligibility. These are patients with advanced metastatic disease that have either progressed on standard of care chemotherapy or intolerant. And we need to have measurable disease, because that's our window of opportunity for a biopsy, to perform whole exome sequencing and RNA-seq to identify those tumor-specific expressed mutations that are put into the vaccine development workflow. That involves the antigenic mutanome workflow that Steve talked about, where we feel like we're doing a really good job of having a higher hit rate and not predicting but validating what are bona fide targets in a very personalized way for a patient. Once that's done, we kind of have our list of what would go into production. In the initial iteration of this, that means that we would have drug substance in the form of synthetic long peptides that are produced that corresponds what we tested at a crude scale within the lab, that are then combined with an immunologic adjuvant, which for the first iteration was montanide, and given to the patient subcutaneously in 3-week intervals with Keytruda. So this was Arm A, 3 shots, up to 5 neoantigen targets per patient for the first iteration of this, and looking for responses radiographically. But importantly, also, since it's a safety and feasibility, where we like to see responses, we wanted to prove that it was safe, and ideally that biologically we're doing what we're saying we're doing, which is this concept of immune editing, letting the immune system go after those particular cancer cells that express that mutation and get rid of them. So how are we doing? So this is patient 1, pancreatic neuroendocrine tumor. And you can see at the top the original version of the trial with the three individual injections with montanide and adjuvant and pembro. Now, at the time, we were limited for production capacity for how many neoantigens we could put into the vaccine, because this is something that we don't have months and months and months to pursue with treatment, particularly for a patient who could be progressing or intolerant of chemotherapy. It is a problem that we are getting better at, and one where we're shortening that time from initial presentation to the cancer center to vaccine delivery. So we had T-cell responses against these 3 mutations. MEN1 is a very important mutation for pancreatic neuroendocrine tumors, as well as these additional mutations in EP300 and this HLA-DQA mutation that demonstrated autologous T-cell responses in the workflow. Now, all these were in production, but at the 11th hour, the MEN1 peptide was not able to be produced. So while we intended to go after it, we could not, but ultimately 2 targets were included in the vaccine. So what you're looking at for this first version with the 3 vaccinations are the 2 mutations that actually were targeted for serial biopsy and our ability to detect the mutation within the tumor tissue after treatment. And as you can see, there's definitely reduction in our ability to detect these mutations, but not surprisingly for tumor tissue that was still there histologically, you could see that there's no effect on a non-targeted mutation, which we believe is evidence that immune editing is occurring. And while we like to see immune editing and stability of disease, we feel like the more you could target, the more doses that you could give, the deeper the response could be. It's kind of intuitive. This is another patient on the first arm. Again, 3 injections with montanide and pembro. And here you can see that 5 different mutations were put into the vaccine, including an MEN1 mutation. So on treatment biopsy, after the 3rd dose, we can see that 4 of the 5, we couldn't detect it. 1 mutation that was targeted, well, we didn't see an impact on that. So these are things we're trying to elucidate, why is this occurring? This patient still has stable disease, and I think would say that this type of immunotherapy is much more tolerable than the alternative forms of treatment that they've been subjected to date. So I think we have these 2 cases of on-treatment biopsies where we have evidence of immune editing. We're also able to look specifically with our ELISpot assay saying, well, we're targeting these particular neoantigens. what happens in the peripheral blood immediately pre and post-vaccine to say, are these T cell responses against these targets augmented? So this is a depiction of targeted peptides within the vaccine, immediately pre-vaccine, with low amplitude cytokine release by ELISpot. And post-vaccine, you can see that for virtually all these neoantigens that were targeted, there was augmentation of T cell responses post-vaccine. So evidence of immune editing, evidence of more robust T cell responses against targeted mutations. What about radiographic responses? So this is a patient with head and neck cancer. You can see here, there's a very large parasternal mass. This is in March of 2019, and here's June. So this lesion, about 90% of this was not detectable after 2.5 months on therapy. Here, we have another cut for the patient. You can see these chest wall lesions here, that were no longer detectable after receiving the vaccine. This actually ended up being a problem for having an on-treatment biopsy. What can we biopsy? I gladly take that problem. So this patient received 3 vaccinations, obviously had a radiographic response. Ultimately, they had recurrence. So this is something that while we did see responses against these lesions, it begged the question, are we targeting enough mutations? Rather than one critical mutation, wouldn't you want to have as many as possible to go after? And should we be giving vaccines longer? And from Steve's work with SCC7, we know the answer is, well, you probably should do that, which led to this current arm of the vaccine trial, Arm C. So what you're seeing a depiction here is multiple sites of injection with series of peptides. Up to 24 neoantigen candidates are able to be incorporated in this vaccine. And we have a new formulation of the synthetic long peptides that are reconstituted and using this viral mimetic, Hiltonol, rather than Montanide. This is the thought of why should your immune system care about a mutation? We all have mutations all the time, but it has to have the right alarm to say, hey, wake up, go after these particular peptides. So it's kind of a bait and switch, make you think that you have an infection. We're loading you up with these neoantigen peptides. Instruct the immune system to go after these and to include Keytruda to prevent T cell exhaustion over time. We did seek a new vendor that's able to produce these in a timely way. So struggling to produce 3 to 5 peptides versus being able to produce up to 24 has, I think, made a world of difference. Multi-pronged attack against cancer. So this is a very different schematic from before, where the Arm C, the patient, is on Pembro every 3 weeks, but you can see where we are doing a weekly prime with up to 24 neoantigen peptides, and then boosting every 3 weeks. And here's a depiction out to 9 doses, so much longer duration, longer, different adjuvant. So let's talk about patient 1 again. So this is after receiving the new version of Arm C, where you can see very large, very well circumscribed liver lesions. Always get paged when giving a presentation. It just happens every time. So you can see that after receiving the vaccine, this particular lesion is much more amorphous. Radiographically is smaller in size. This lesion actually, in its longest dimension, is measuring a bit bigger, but you can see it's kind of faded out, not as well circumscribed as before. So the majority of these lesions that were measured actually got smaller as a function of the vaccine. This had not happened before with this patient. Here we see another depiction of another portion of the liver, where this particular lesion is also getting smaller. So we were able to pursue biopsies, ultrasound-guided biopsies of some of these amorphous lesions, one that was definitely responding versus one that maybe was responding, maybe not. The one that looked like it was intermediate still had detectable neuroendocrine tumor in it. The second one had, despite, I think, 3 to 5 core biopsy passes, no evidence of the neuroendocrine tumor. What it did see was normal liver tissue and macrophages. So we think that the right stuff is where the tumor used to be. Obviously, we're very interested in profiling the remaining tumor tissue to say, well, what happened? What's the mutational landscape of that biopsy? Is there evidence of immune editing like we did before? But more importantly, given more time, will we see additional regression because this patient's not done receiving the vaccines because we're able to do more longer. part of our immune monitoring program that I'll touch on briefly is while we're happy to see mutations go away that are targeted radiographic responses, believe me, we're happy about that. We'd also like to know what are the T cell receptors that are driving this? Can we detect them? Can we clone them? Can we use that as another method of monitoring for effective T cell responses as a function of time? So what you're seeing here is where we're able to look within the blood of patients when they do this 2-week assay with the relevant neoantigen targets and look for expansion of various T cells within that culture, presumably because they like what they see. This is a neoantigen that there is a T cell response, and given appropriate in vitro culturing, they expand, sometimes 2,000-fold. This is looking at cytokine release assays where we are able to determine the frequency as well of or single cell sorting of these cells, that we can see the variant or the frequency of the beta chains, where we can sort these cell by cell and then use paradigm sequencing to say, well, what's both the alpha and beta chain for each of these T cells? And we have an ability to take those when we know what are these T cells that have expanded most in the culture, to be able to put this into a expression system with the beta, P2A, alpha, T2A, and some marker, in this case truncated CD34, and introduce those to a surrogate cell, J76, or even PBMC from the same patient, allow for expression of that T cell receptor, and autologous to the patient, take their blood and have immortalized B cells that work great as APCs, and either pulse it with a relevant neoantigen peptide, a relevant peptide, or just use in vitro transcribed RNA to see if this can be naturally processed and presented rather than peptide, and say, do you see your target? So we've done this. This is an example of a patient that had peripheral responses against KRAS, which is a bad mutation to have in colorectal cancer. And as you can see, for these 3 individual T cells that I'm depicting, although more were identified, there are specific responses against these APCs, in this case pulsed with the peptide, with the TCRs that are independent against each of those. So this is our ability with sequencing to see what's happening for immune editing, and to clone these and to see what we want radiographically and find the T cell receptors that are responsible for it. So I'm happy to share that information with you. As Steve talked about, we do have a platform that we think we're finding things at a higher hit rate. It works with these low mutational burdens that have been not allowed to have a lot of these IO therapies. And this personalized cancer vaccine trial is underway here. And here's my version of the slide. So Martin was critical for the T cell analysis. And yes, don't mess with Malad. Thank you very much.